Okay, and we have spent a lot of time talking about the tags, and we hinted something about the gene regulation. In the next maybe 20 minutes or so, we'll keep continue that line. Right? Sometimes it's really hard to distinguish the technologies and observations and their impact on the gene regulation. I'm trying to do my best to separate the lecture, but next one again, they are pretty closely related. Okay, so this again, this figure is from the original Science 2009 paper. Again, this is the chromosome-wise interactions, 14, right? Again, each red spot meaning there's strong chromatin actions between this region to the other region, right? To make things look better, right? So essentially, for each diagonal, you can compute a average, right? So you can see the expected value for here will be really small. The expected value for here will be really big or strong, right? So figure B, essentially they just use a observed value divided expected value. So, and then if you take a log, you can see the more clear um, uh, difference between positive and negative, right? You can have a red and blue. Figure C is really interesting. You can see the pattern is much more clear, right? So what they did is they compute for each cell, not the real cell, each cell on the data matrix. They can compute Cij. You can compute the correlation between the ith row and the jth column. Right? For this spot, same thing, you compute the ith row and the jth column. So what does this correlation really tells you, right? So essentially, if the two cells are close to each other, it's not surprising their interaction profile across the whole chromosome are similar. So these two are red. But what is really surprising is actually this spot. That means the location here and the location here. Although they are pretty close, they are far away in the genomic distance, their interaction profile across the whole chromosomes is very similar, right? So what does it tell us, right? How do we analyze this data? Right? Again, I think you have covered the PCA analysis, or you will cover PCA analysis. Right? Essentially, PCA just gave you the dimension reduction, right? So in this case, we can use PCA analysis to reduce this high C matrix into this 1D eigenvalues, eigenvectors. Here, just what we show is only PC1, right? So for PC1, you can see the positive values and negative values. Amazingly, you can see the PC1 values and the positive and negative values. They are really corresponding to the checkerboard patterns in the genome, right? And when the authors, they compare the eigenvectors with the gene density, with the homomorphism marks, they observe actually the positive values, which they call compartment A, is really corresponding to the gene dense regions and a lot of the active transcription values. And the negative values for eigenvectors, they really corresponding to the gene desert. And a lot of times they're enriched for repressive marks. So one thing you can observe is actually potentially the A compartments, right? They still interacting with A compartment, even if they are pretty far away from, they are pretty far away in terms of the genomic distance. Right? And the B blue part time, the blue bars, they are also interacting with the blue regions. A interacting with A, and B interacting with B. So what again? We are talking about cells. What does it really tell you, right? Again, so this is a very nice review written by Job Decker last year. Again, in the beginning, you have your cell. This is cell nucleus, different chromosomes or chromosome territories. And essentially A and B. A is more active, means they are relatively closer to the inside of the cell. And compartment B, they are closer to the outside, to the nuclear envelope, to the lamina, meaning B is more repressed, right? And the idea is because all the A's are really inside the nucleus, they tend to interact, because once you imagine the cells are folded, so A are more on the inside, so the A tend to interact with A compartment, even if they are pretty far away from each other. And B is similar story. So that's why you begin to see these checkerboard kind of patterns, right? Again, only we, or the authors in that paper, they only define A and B compartment essentially based on strong positive and negative values, right? Which is good. But as a mathematician and a computational biologist, you can imagine, oh, sometimes there's strong enrichment, and sometimes there's really, really low negative values, right? Maybe there are, you can put them into a different degrade, right? So further on, I think there's a Rao ETL 2014 and a few following up papers. They began to further dissect the A and B compartment to subcompartment. So they began to say, okay, there's besides A and B, there's A1 and A2 compartment. There's a B1, B2, B3 compartment. The potential hypothesis actually A1, A2, and B1, 3, B2, they are interacting with different partners, right? So in this case, you can see again, A is more in the center, and those regions are more open and then more transcriptionally active, right? So you can see in A, they are the compartment closer to the nuclear speckles. There are a lot of transcription going on. And A2, they are more PAL2 and Bruno domains in this region. Again, but both A2, A1, A2, they are open compartment and actively transcribed region. So for B, because there are different repressive mechanisms, right? For B1, it has been proposed, there are a lot of the polycomb proteins. There are many polycomb proteins, they come together, sort of like have their own phase separation kind of mechanisms, and all the DNAs will come over surrounding those polycombs and really repress the gene expression. And the B2, they are pretty close to the nucleosis. B3 is more on the edge. They are closer to the lamina, and HP1, they're enriched for HP1 protein, right? Again, to further dissecting the genome into different compartments is still an actively studied field. And there's still space for you guys to do some damage in that field as well. Okay, so previously we talked about tags are pretty consistent. So how about compartment, right? It's quite amazing. In this case, again, we are talking about the same 5 cell types, ESL, and the 4 different cell types. Again, if you only see the tags or the high-C interactions, they are pretty much the same, right? But once you do your conversion, you calculate their eigenvectors, you can see their compartment is quite different. You can see these 5 cell lines, high-C are very similar, but compartment, you can see there's a B compartment, A compartment, B compartment. It's quite divergent. So it seems like if the compartment annotation, or at least the PC1 eigenvectors, they possess more diversity, right? And then you may ask, so whether the A and B compartment, they will have any effect on the gene regulation, right? I will use one example in our lab. In this work, all right, we have 2 conditions. You can imagine condition 1, condition 2, where we did CRISPR, right? So you can observe in this case, this is a B compartment, this is A compartment. And these 2 purple tracks are the ChIP-seq for H3K27 isolation, right? And then the last part is RNA-seq. What we're investigating is it's a leukemia gene, PON2 gene, right? So in the normal condition, this region is B compartment. But once we did our CRISPR experiment, this region becomes an A compartment. And as a result, the expression for this gene is upregulated by almost 5-fold, right? And then when we further compare what we observed in the cell line in the patient, again, so this is high-C data. In this case, you can see these few guys, this patient and cell lines, they are A compartment, and these are B compartment. Again, we observe this gene is highly expressed in the A compartment rather compared with the B compartment. And overall, when you manipulate the genome, you can, in general, the genes, when they are located in the B to A switch, their gene expression is really upregulated. And when the genes are located in the A to B compartment, their gene expression is really reduced on a global level, right?